Hello, and welcome to this week's Ethical Dilemma in Science, where we're going to take a look at zombie pigs, preserving life and some brain functions after death. So, with all of these ethical dilemmas in science, they're based, at least in part, on some type of recent news events, and this one is no different. In August 2022, a number of popular press articles available talking about kind of this almost like resurrection, or at least kind of preservation of life in a pig after it had been dead for a period of time. And so I dug around, found the actual scientific article, and that's here, Cellular Recovery After Prolonged Warm Ischemia of the Whole Body. It was published in the journal Nature in 2022, August of 2022. And so what they did in this study is basically anesthetize a pig, induce a cardiac arrest, and allowed the pig to basically be at cardiac death, so no heartbeat, no circulation, for a period of a time of about an hour, actually is an hour. And so during that hour, this is what the warm ischemia is. There was no blood flow, so no oxygen and nutrients being distributed through the body. And so it wasn't hooked up to a heart-lung machine. They weren't doing any artificial perfusion. They weren't doing any CPR or anything like that. And then they hooked the pig up. So the pig had been dead for an hour. They hooked it up to a series of pumps and filters and circulated this special cryoprotective blood substitute. And what they were able to show is that over a period of time, they were able to kind of decrease the amount of cell death that would normally be occurring. They were preserving tissue integrity, so preserving the structure of the cells, and in some cases restored cellular function and molecular processes. And so, it's important to recognize that even though some of the popular press articles were talking about this as kind of resurrection of the dead animal, it was not really doing that. It was just basically preserving and restoring some cellular function. But ultimately, even though it's not clinically relevant now, it may have some clinical implications in the future. And so kind of getting into kind of researching this article, I found out that the same group had published an article in 2019, which again is kind of really, really interesting, at least to me. What they did in this study, and this is a 2019 article, and I didn't write down the journal, it's again in the journal Nature. What they did was obtain decapitated pig heads. So pigs that were sacrificed for food products in a slaughterhouse. They went and got the heads after 4 hours, where they were basically, the decapitated heads, the brains were not hooked up to anything for oxygen, not getting any nutrients, not anything. They hooked up these decapitated pig brains, again, to a similar series of pumps and filters with a similar fluid. And then for a period of time, so kind of hooking this up after 4 hours of kind of death and no attempts to kind of preserve function, they were able to detect some basic levels of brain activity. So it's important to recognize that it wasn't like neuronal signaling, it wasn't kind of conscious perception or cognition, anything like that. But this is a very important study because it shows that the brain, potentially, at least basic brain function, is more resilient than what we had originally thought. And there may be ways in which the brain can survive for longer periods of time. And so if we take a look at this, again, this kind of brought to mind Mary Shelley's Frankenstein from 1818 and the classic 1931 movie, where you've got a scientist that is attempting to, create life from death. And so in the movie, Dr. Frankenstein basically kind of cobbles together these dead body parts and like a modern Prometheus, kind of restores at least life into Frankenstein's monster. Now, I've always used Frankenstein in one of these ethical dilemmas in science relatively recently, so I wanted to go to a different. So instead of looking at Dr. Frankenstein, basically going with a different movie to reference this, so this is The Brain That Wouldn't Die from 1962. Just look at a short kind of series of clips from this.
I should have known he was as good as dead when they wheeled him in.
You did everything possible, everything you could, Dr. Gould.
Everything? Everything except save my patient.
Now, Dad, do I have permission to take over and try things my way?
The operating room is no place to experiment.
He's dead. I can't do any harm.
Pulse is coming back stronger than ever. It's unbelievable.
Nothing's unbelievable if you have the nerve to experiment.
At this point, the doctor's fiancée was decapitated in a car accident and was going to try to preserve her.
My eyes are deceiving me.
What you see is real. What's done is done, and what I've done is right. It's the work of science.
Okay, so the article, or at least this movie, was written 60 years before the article and before these research studies were done. But it does raise the question, how do we study the brain? How do we study function associated
I should have known he was as good
with
as
the
dead when they
nervous
wheeled him.
system? So one of the easiest ways to study the brain is the process of tissue culture. And so in tissue culture, in many cases working with embryonic animal tissue, so this is an example of a rat embryo here, they dissect out the brain and then from the brain isolate individual cells and then grow the cells kind of in a laboratory setting, grow them into, in essence, plastic petri dishes, or in this case, little wells that are present. But basically a little microenvironment where we can control the kind of conditions that these cells are living in. And so you can look at individual cells. In some cases, you can mix cell types together and look at very simple processes. And so these are kind of nerve cells that are growing out. You can look at it under higher magnification and study some of the basic processes. And so there are a lot of studies that are done in tissue culture looking at kind of isolated cells. But these isolated cells are really in an artificial environment. And you're not seeing the kind of natural interaction that's going to be occurring between these cells. And especially when you're looking at the brain, some of these interactions are going to be important for understanding the actual cellular interactions and processes that are occurring. So another way that the brain can be studied is through the process of tissue slices. And again, using an animal model, basically dissecting out the brain, kind of taking very simple slices through that, and then taking those slices and put them in the equivalent of one of those petri dishes. Put them in the equivalent of a little plastic dish where we're able to add, specific ingredients, specific solutions, like the solution that was circulated through the pig in the nature studies that started out this talk. And we're able to kind of preserve the cells that are together in that slice of tissue, preserve some of those interactions that are occurring for a series of a couple days. And you can go in, and this is looking at kind of putting in little electrodes and kind of measuring the electrical activity. But again, this is still an artificial environment. This is looking at kind of small regions, micro regions within the brain, and you're not seeing kind of the greater connectivity and the interactions that are occurring between the nervous system and the rest of the body. More recently, some studies have looked at creating brain organoids, and so these are kind of like artificial brains that are grown in culture. And so basically, these are working with some type of embryonic stem cell, in many cases, working with animal cells that have been isolated or created in some way, growing them under specific circumstances, so again, in an artificial environment, but they're able to develop, again, in this artificial environment, so an artificial brain, a brain organoid, and you're able to look at kind of broader connectivity, broader connections in these artificial brains that are occurring. And many of these studies have been, kind of looked at over a short period of time. More recently, they've been able to do this for extended periods of time. So in this case, they're looking at it upwards to about 6 months. And again, this is a great model system, but it's still inadequate for looking at some of that greater connectivity that's going to be occurring between the brain and the rest of the body. If you want to be looking at drug effects, you want to have an idea about how the drug is going to be kind of absorbed within the body, how it's going to be distributed through our bloodstream, and how it's going to get to the brain and have a function on that. Now, these brain organoids, we did have an ethical dilemma in Science earlier this year, and I'll link to this in the comments below. Human brain organoids, enhancing a research model and or creating a conscious entity. So again, it's a better model for some types of research, but it still doesn't give us that connectivity. And so then a question becomes, is it possible to study the whole brain? Is it possible to study the whole brain in vivo? In vivo basically means in life. So can you study the brain kind of in the body? Can you study it in a living organism? And again, looking at kind of animal models here, we've got a rat. We've surgically gone through and implanted in some type of electrode. Other times you can implant in something that's going to deliver drugs into that region, so you can look at the effect of the drug in that region of the brain on the overall functioning of the animal. And again, that is a good model system for studying different properties, but it becomes a little bit more difficult if you want to study kind of broader interactions, if you want to study kind of at higher levels. And so if we take a look at this, we really want to try to understand how the brain is going to be operating and working in the human body. And so we're going to get to another kind of ethical dilemma. This is a study that was published in July of 2022, in which they were basically using a dead body to take a look at kind of the kind of response of transplanting a pig organ that had been genetically modified to kind of be better accepted by the body. But they basically implanted that into a brain dead individual with a proper consent, proper ethics associated with it. And they were able to study it for a period of time where they could go through and do biopsies of the tissues. They could go in and look at the response over an extended period of time, which they wouldn't be able to do in a living recipient. And so in this case, the study was terminated after about 3 days. And so they, took the basically subject, the dead body that was preserved under a life support system, and then terminated the study at that point. Again, this gave rise to another ethical dilemma in science from earlier this summer, post-mortem research, a critical model for biomedical research, and or mimicking Dr. Frankenstein. This is the one where I referenced the Frankenstein movie. And so, again, lots of ethics that are involved in these studies. They were all done properly, but it does raise the question about how this can be done and how this can be used in the future. And that gives rise to the ethical dilemma in science for this week. If we were to use this system of preserving the body in a much more kind of effective functional state than what's currently possible with the current heart-lung machines and processes that we have available, how does maintaining some level of brain activity after death affect its use in research? What type of protection should be in place for post-mortem brain research? Right now, the current guidelines are focused on preventing or minimizing pain in living organisms. But once that organism is dead, what does that mean about the protections? What needs to be considered under those circumstances? Would the protections or would the processes be different if the scientists were able to restore very simple neuronal signaling? They used blockers in the study right now to limit the brain activity, so they were able to look at just simple basic cellular function within the brain. Over time, improved ability to kind of fine-tune the system, potentially we would be able to, create or at least restore some level of consciousness within this non-living organism, kind of the brain that wouldn't die. In essence, the movie from 1962. And when we're looking at this, we're focusing on this within an animal model. These studies were done within a pig. But what happens in the future? There is a possibility that with proper informed consent, this could potentially be a model for studying the human brain after death. So for individuals that donate their donate their bodies to science, you may be a specimen in a cadaver lab in a medical school. and the body would be studied for a year before it's going to be retired, it's going to be cremated, and the remains interred in a respectful ceremony. Is there the potential that we would have these individuals that have essentially died that could be used as a test model for some type of biomedical research in a human situation? And so that's not now, it's not even in the near future, but this is something that we need to be thinking about as this technology develops. So as always, please see the description below for additional information and references, and I'll link in those 2 other ethical dilemmas in science in case you're interested. So thank you for listening. We'll have another one of these ethical dilemmas in science next week. Have a great week, and hopefully see you soon.